Thanks everyone very much for attending today. I'm very proud to present Adam Cisterne to you all. Adam has spent the last few years doing a really impressive job on a really difficult bird, the Tasmanian Mast owl. Despite meeting some really major barriers thanks to COVID-19, in addition to the general barriers that mast owls themselves threw in his way. He's done a really great job. And I'll hand over to Adam now to present the findings of his last few years of fantastic research. Take it away, Adam.
Okay, well, I'll introduce myself again. I'm Adam Cisterne. I'm a PhD candidate with FENA, and I work with the Difficult Birds Research Group, supervised by Dr. Dejan Stojanovic, Dr. Ross Crates, and Professor Robert Heinzen. And yeah, I'm here to discuss my PhD research. And the title for today's seminar is Conservation in the Context of Prevailing Data Deficiency, the Case of the Tasmanian Mast Owl. So human activities are impacting other species. Abundance of all orders of organisms is down and extinction risk is growing. 28% of species are defined as globally threatened, and coinciding with this loss of biodiversity is the collapse of many of Earth's ecosystems. Conservation requires data, for example, on population sizes and behavior. Unfortunately, there are over 20,000 data deficient species on the IUC red list alone that simply can't be assessed. At national, regional, and local scales, threatened data deficient populations nevertheless require conservation intervention, but achieving this is difficult. Now, for many populations, data deficiency persists simply due to intrinsic barriers to gathering data. For example, there can be high uncertainty about approaches to surveying or detecting a species. Small population sizes, low densities, and cryptic behaviors all reduce sample sizes, and these barriers can entrench data deficiency. Data deficiencies delay assessments and stall recovery. Recognizing data deficiency, therefore, can attract investment in research. However, as these species are difficult to study, research programs need to be resilient to setbacks and avoid complacency and indecision. And this set me on a pathway of thinking about how best to reduce data deficiency whilst achieving conservation outcomes in spite of knowledge gaps. So I began by building up a generalized framework for working with data deficient species in the context of conservation. Given the magnitude of the knowledge gaps for these species, the obvious first step is new research to reduce data deficiencies. But there can be a dizzying array of unanswered questions and hot debate about their order of importance and the best way to research them. Acquiring new data is invaluable because it is contemporary and relevant for answering targeted questions. New field data could include evidence from surveys, Indigenous expert and local knowledge, but no one study can answer every question, and so multiple studies are needed, which can be difficult and costly to achieve. Also, for rare species, small sample sizes can hinder inference, so there are limitations. Existing data, for example, from collections, data repositories, and the literature can be easier to obtain, can contain substantial sample sizes and an historical context. However, it can be biased by its opportunism, which can limit its scope. And again, for rarer species, these sources can also be scarce, so there are limitations. A research plan that incorporates a range of methods from new and existing data sources can be resilient to the difficulties of studying rare and poorly known species because 1, it can adapt to failure. 2, data sources can supplement one another, boosting sample sizes. 3, alternative sources of data can target a broader range of questions. And 4, as information becomes available, there is more opportunity to improve study designs for the species. A multifaceted adaptive research plan may have a better chance of reducing data deficiencies. Still, The insights that we gain may be less than expected and data deficiencies will persist. Even so, in the context of conservation, managers must decide how to act, and so approaches to making decisions in the face of uncertainty are important. Quantifying the effects of uncertainty could give us confidence in our assessments and advance conservation research. This could help also to identify concealed threats and possibly lead to actions. I studied the endangered and elusive Tasmanian mast owl as a model data deficient species, aiming to reduce data deficiency and improve conservation planning through an adaptive research approach. Only limited life history and other population are available, and the effectiveness of current protections are unknown. My thesis adapted my generalized framework for working with data deficiency by targeting 4 broad aims exploiting different data sources and methods. These aims included attempts to gather new data asking 1, how widespread are Tasmanian mast owls in forests? 2, can new sources of field data increase opportunities for research? Then I complemented these with with existing data sources seeking to ask, 1, is the population inbred? Has it declined? Is it one or multiple genetic lineages? And 2, how will the Tasmanian mast owl's distribution be affected by climate change? In my 5th study, I take a management perspective to explore the effects of uncertainty in data and knowledge when making quantitative assessments of threats to the mast owl. Although my experiments were not all successful, my plan allowed me to adapt and repurpose data and knowledge to increase the value of each successive study. So now I'll take you through each individual study. Mast owls have been surveyed twice in the 2 decades prior to my arrival, and their occurrence was predicted by mature forest and lower altitudes, typically below 600 meters. This places the population in direct conflict with logging. Therefore, we aimed to understand how widespread mast owls are in logged forest and why they use some locations and not others. This first chapter was published in Austral Ecology. The work was funded in part by the Stuart Leslie Bird Research Award, and thanks also to the Forest Practices Authority for their support. So for those who are not familiar with occupancy models, they estimate occurrence from detection histories. Sites are surveyed multiple times. Presence is predicted with respect to detectability. It's a good option for rare and elusive species where detection is not guaranteed even when individuals are present. These models partition site-level variables, such as the number of tree hollows, into occupancy, and survey-level variables, such as wind noise, into detectability. I sampled 800 square kilometres of logged forest in Tasmania, south of Hobart, shown in this map. The map shows the sites surveyed, with Hobart just off to the top right. In fine weather, we surveyed 160 small sites, minimising the survey time to 10 minutes and maximising repeat visits, making 4 to 5 visits within a 9-week period. This dense but highly replicated survey effort had been shown to increase detections of other elusive species whilst limiting within site level heterogeneity of habitat features. We used call broadcasts to elicit wild mast owls and recorded mammalian prey with a thermal imaging scope. I did a total of 662 surveys. I made 29 mast owl detections at 19 sites, and they are indicated here by black filled dots. On the left are site level variables, which included a variety of habitat and landscape features. Most importantly, mean prey abundance was calculated as the total count over all visits divided by the number of visits. And on the right, the survey level variables included moon phase and temperature. We limited our models to a single main effect in occupancy and detection to avoid overfitting a very sparse data set, and ranked them by AIC. The top model included an effect of mean prey abundance in occupancy and constant detection probability of 0.26. The figure shows models that ranked within delta AIC5 of the top model and the null model at the bottom. Habitat and landscape variables did not rank higher than the null model. And two interesting results came from this study. The first was that masked owls may be using sites with a greater abundance of prey available. However, detections of prey and of owls were overall rare, and predictions from occupancy from this data had very low confidence, shown here in the figure on the left. Second, on the right, we have a spatial correlogram of owl detections. Owl detections were spatially autocorrelated in short distance classes of 1 to 3 kilometers, which, given the large size of home range predictions, suggests that the same owl or pair of owls occurred in nearby sites. Considering that so few detections were made in between and the survey effort was substantial, we concluded that very few owls persist in the study area. So occupancy models do not perform well with detection probabilities below about 0.3 when naive detections are also few. Our single visit detection probability of 0.26 is low, and detections occurred in fewer than 5% of surveys. Consequently, the cumulative detection probability shown in this figure was well below 1 after 4 to 5 visits, and therefore the occupancy estimates remain imprecise. In our case, increasing the number of visits to each site will only mildly increase the cumulative detection, and therefore is probably not worth the extra effort. These techniques did not yield robust conclusions for me despite a large effort, because detection probability was low and detections were rare. Greater survey effort was unlikely to provide further insights into masked owl occupancy. Rather than get stuck, I decided to move on from these techniques. New techniques and new sources of data can be a game changer for the study of rare and elusive species. Call broadcasts did this for the study of nocturnal birds. DNA has done this for biology in general. Therefore, I decided to investigate new sources of data for researching masked owl ecology, hoping that I might increase detections and increase modeling opportunities. Which brings us to chapter 2, SMP markers from feathers and pellets for population genetics and individual identification. This work was funded by the Holsworth Wildlife Research Endowment, and special thanks to Trewanna Wildlife Sanctuary and Nature World for their help with this project. So wildlife leaves behind a diverse range of signs that they've occupied an area. Traces indicate a location, and the DNA identifies individuals. This opens the door for more detailed studies of species spatial ecology, such as density and abundance estimates, and for non-invasive genetic sampling. Trace surveys do not require individuals to be present at the time of surveys, and they maximize the information gained from each new detection. Therefore, they can be a powerful tool for studying species that are otherwise difficult to survey. I investigated whether feathers and regurgitated pellets were a reliable source of DNA for studying masked owls. I devised a small experiment to determine 1, the success of DNA extraction and genotyping, 2, the genotyping error rate, 3, whether unknown samples can be matched to their origin, 4, whether data are useful in population genetics, and 5, whether data are suitable as individual genetic tags. For this experiment, I collected molted feathers and pellets from owls in 3 scenarios, and DNA was obtained from pellet surface swabs and from feather shafts. In the first scenario, we ask if known owls could be matched to their pellets and feathers. Feathers and pellets were collected from 3 owls housed separately. Therefore, we had known trace DNA samples from known individual owls. In the second scenario, we ask if random pellets could be matched to a known owl. The same 3 owls were housed together for a period, and a collection of pellets was made on a single occasion. Therefore, we had random trace DNA that had originated from these known owls, but we didn't know which. In the third scenario, we aimed to differentiate among unknown owls from within groups of randomly collected pellets. Feathers and pellets were collected from piles produced by unknown numbers of captive and wild owls. SMP loci were obtained through a DartSec protocol, which used a separate cohort of high-quality mast owl tissue, N equaled 56, from my next chapter, so that SMP were representative of the population. The initial dataset was filtered for high accuracy, and the final dataset had 1,042 loci. The successful sequencing of SMP in trace specimens was low at 38% of all collected specimens. However, this is comparable to that from published data from microsats, also in our pellet swabs. Missing loci per specimen ranged from 1.3 to 85%, indicated here in the red rectangle for the successfully sequenced traces, and was not different between freshly collected material or material of unknown age, here in the lower right box plot. Due to this high rate of missing loci, any two specimens only overlapped at a subset of the SMP, on average at 60%, which from here I refer to as shared loci. So, to the results of scenario 1, which had known owls and known pellets. Pellets and feathers from the same owl matched at 98.5% of shared loci, seen as red squares in this figure, whereas pellets and feathers from different owls matched at only 81.5%, seen as bluish squares in this figure. This indicates a genotype discrepancy of approximately 1.5% in individuals. To the results of scenario 2, which had random pellets from known owls. We added the pellets from scenario 2 to the results of scenario 1, indicated here in the black rectangle. Two randomly collected pellets from this communal cage matched to owl C at approximately 99% of shared loci versus 80% to other individuals. Therefore, these pellets were very likely from owl C. The results of scenario 3 are presented alongside all other data from the first two scenarios. I calculated the pairwise genetic distance between all specimens from all scenarios and mapped them onto a neighbor joining tree. Based on this measure of genetic distance, specimens originating from the same individual are grouped on shallow branches. In contrast, deep branches connected unrelated specimens. To the results of scenario 3 specifically, the red rectangle indicates a wild roost site and the blue rectangle an enclosure. All of the pellets and feathers from the roost site with an unknown number of owls grouped on shallow branches, and likewise for the pellets collected from the enclosure. When contrasted to the results of other scenarios in this tree, the branching pattern suggests that all specimens within the red and blue boxes are attributable to individual owls. Therefore, the genetic relationships between specimens are defined by a common population genetics metric, genetic distance. We were confident that these SMP could define the population and individuals. Next, we wanted to find the smallest subset of loci that could identify all individuals. We filtered down to a set of 156 SMP using high population heterozygosity and low population FST. Based on these 156 SMP, there was a negligible probability that an individual's genotype could match a randomly drawn unrelated individual. This was pretty good, and 21 SMP could confidently identify every individual in a population of 2,000 owls, which is reassuring given the high missingness in the data. So trace-sourced DNA was of sufficient quality to provide usable genotypes, with the major limitation being sequencing success rate, which can only be overcome by collecting large amounts of material. The potential of pellet-sourced DNA opens the way for systematic genetic sampling, and genetic tags open the way to use DNA to estimate density and abundance. However, we need to be able to systematically search for pellets in the landscape for their potential to be realized. At this stage, I engaged in approximately 100 hours of searching with a scent dog trained on Mast owl pellets. However, we located no pellets, even in areas where owls were at least detected in the evening. Therefore, the rarity of these traces in the landscape may still be a factor that limits this kind of research. After 2 field-based surveys, I realized that the sample sizes that I required to do further modeling, specifically of bird densities, or to start to look at population genetics, were unlikely to be obtained in this way, at least within the time available to me. Therefore, I sought other data sources to address the following chapters. Estimating population sizes from field data and assessing connectivity through demographic studies are very difficult for low density and cryptic species. However, genetic methods exist for studying both if a sample is available to do so. Which brings us to chapter 3, genetic structure and diversity in the Tasmanian mast owl. This work too was funded by the Holsworth Wildlife Research Endowment. So population declines have genetic consequences. Therefore, genetic diversity and its distribution is vital information for the conservation of species. In this study, I sampled DNA from a natural history collection. For temporal analyses, we group historical specimens from pre-2000 and contemporary specimens from post-2000, indicated here in the figure. SNPs were sequenced by a DARTSec protocol and filtered for quality and coverage. I looked at 2 important population genetics topics: 1, genetic diversity and effective population size, and 2, population genetic structure. So to part 1, the loss of genetic diversity is a substantial threat for many species as population fitnesses and adaptive potential may decline. The effective population size is an indication of exposure to genetic drift. Declines in effective population may indicate bottlenecks, fragmentation, and declines. We calculated observed heterozygosity, expected heterozygosity, and inbreeding coefficients for both our historical and contemporary subsamples and compared them with Mann-Whitney tests. We found no significant differences between these measures of diversity between the time periods. FST was low at 0.002, but there was sufficient power to say this was significant, shallow differentiation between the time periods. We calculated effective population sizes also in the historical and contemporary subsamples using the linkage disequilibrium method, adjusted for overlapping generations using 3 methods that account for MB to NE ratio, adult lifespans, age at first breeding, and variations in breeding. And the 3 adjusted values are shown in columns 3 to 5, indicated in this red box. We found that the estimated range of effective population sizes for the historic subsample were higher than for the contemporary subsample, which together indicates potentially a declining effective population size. Now to part 2. Genetic structure provides us with an idea of the scale at which genetic diversity varies within and among populations. It informs us about gene flow and genetic differentiation. These are important concepts in conservation for defining management scales, which both capture genetic diversity and promote the retention of it and continuation of gene flow. In this part, we explored spatial genetic structure. We looked for genetic clusters using 2 methods. We first ran a principal components analysis, shown here in the top figure. Individuals mixed well on the first 2 principal components, with only a minor spread on the x-axis coinciding with latitudes. Discrete clusters are not visible. We also found no evidence of genetic clusters with a discriminant analysis of principal components, and together These suggest a single genetic population. We then looked at whether the genetic relatedness of individuals changed with respect to distance. We tested for isolation by distance using the Mantle test and found significant isolation by distance across the sample, shown here on the left. A spatial autocorrelation test found that individuals within 60 kilometers of each other are more related than expected, whereas relatedness becomes weak beyond the 140 kilometer distance class, demonstrated here as a spatial correlogram. Both analyses support a model of isolation by distance. The eigenvalues of a spatial principal component analysis carry information about both variance and spatial autocorrelation. Here, the squares represent the location of each sample, colored by the lagged score, which smooth the eigenvalues for better visualization. Genetic variation is correlated here in space. That is, nearby samples are more related than distant ones. A global pattern of genetic differentiation is clearly represented, as the most northwest and southeast samples are the least related. Interesting, though, that the genetic variation appears more strongly correlated across the north and less so across the southern valleys. Although genetic diversity did not change, evidence of a declining effective population size raises the possibility of deterioration between the 1970s and 2000s. From our NE results, we estimate an adult population size of approximately 1,800 prior to 1970 and approximately 700 in the first decade of the 2000s. These estimates appear to validate the original assessment of the population status as endangered at the turn of the millennium. We did not find multiple discrete genetic lineages, which suggests the population is a single conservation unit. However, we found significant spatial variation, probably from isolation by distance, suggesting that mast owls do not disperse long distances compared to the size of their range. Limiting the impacts of threats within neighborhoods will be vital to conserving genetic diversity, which may require large reserves of intact habitat to ensure that dispersal and settlement can continue at that scale. Defining management units can be complicated by continuous population structures. Therefore, a careful range-wide policy of protection should aim to support the population's global diversity. This work demonstrates the value of existing resources to improve data and address critical questions in conservation. Our results also highlight the power of population genetics to reveal processes that are otherwise challenging to detect in cryptic species. Next, I wanted to look at broader level patterns for the mast owl and the ecosystems in which they live during a heating climate. This was of interest because climate change is likely to negatively impact many species. However, this has not been looked at for the mast owl. This takes me to my fourth study, where I zoom out from individual and population processes to range-wide distributions of this predator and its prey. To investigate this further, I utilized my own data and citizen science databases such as iNaturalist and others, which can contain species occurrences in sample sizes that are pretty unimaginable for rare species. Which brings us to chapter 4. Climate change impacts on prey distributions may constrain the range of the Tasmanian mast owl. At macro scales, species distributions are strongly affected by climate. Therefore, climate change may drive shifts in ranges. However, research now predicts that distributions are also affected by habitat and by species interactions. These interactions can be disrupted during climate change, and range shifts can have cascading effects. Predator and prey richness co-occur geographically. Predator distributions can be affected by the impacts of climate change on prey. The mast owl is a generalist, and prey-rich ecosystems may allow generalist predators to exploit broader than expected climates. I used two species distribution models to investigate the effect of climate and prey availability on mast owls. A bioclimatic model defined by only climate variables and a biotic model defined by the same climate variables plus prey richness. And it was achieved in this way. The mast owl bioclimatic model was defined with a subset of bioclim variables for the current climate, and final predictions were made with ensembles from multiple models. This was repeated for each of 11 prey species, and those predictions were summed to obtain a single layer for prey richness. Then the mast owl biotic model was defined by the same bioclim variables, plus I added in the prey richness layer. The performance of both models was comparably good, indicated here by high AUCs and very similar ROC curves. The predicted distributions are similar. However, the biotic predictions capture a broader area. Overall, mast owl distribution was predicted by lower precipitation, mild to higher temperatures, and greater prey richness. Next, I predicted mast owl distributions under various climate change scenarios. We used 3 global climate change models, each in high and low representative concentration pathways, and 20-year periods centered on 2070 and 2090. We obtained future climate data then for 12 predictions in 3 scenarios, that is low and high RCP in 2070 and 2090 for 1, a least hot, least dry future, 2, a maximum consensus future, and 3, a hottest and driest future. A prey richness layer for each future climate was made by predicting and summing prey distributions using the modeled future climate variables of each scenario and the original prey SDMs. Then mast owl distribution was predicted with the bioclimatic and biotic SDMs using the modeled future climate variables and future prey distribution in each scenario. First, to the least hot, least dry future. The results show that in a least hot, least dry future, here in the red box, mast owl distribution moved further east and retracted from the north and the south in both bioclimatic and biotic predictions. This future climate has a higher upper range of precipitation than the current climate, which may explain the lack of contrasted observed effect. This same pattern is seen in predictions of the biotic model in both maximum consensus and hottest dry scenarios, indicated here in red boxes. In contrast, in these scenarios, predictions of the bioclimatic only model indicate a possible expansion of mast owl distribution across the entire eastern half of the island. So this figure shows the gross change in area predicted under climate change compared to the current mast owl distribution from each model. The bioclimatic model predicted a loss of area of potential distribution in the least hot, least dry future, indicated there in a red box, but a gain of area in both maximum consensus and hottest, driest futures, both represented there in black boxes. The biotic model mostly predicted significant losses of gross area in scenarios, indicated there in a green box. The mast owl seems quite resilient to climate change, or at least to drying climates. However, prey richness contributed strongly to the prediction of mast owl distribution. Therefore, under some scenarios, the impact of climate change on prey may restrict the distribution of mast owls. Now, our models did not consider the abundance of prey or relative importance of different prey species in the mast owl diet. Abundant prey may allow the owl to exploit less prey-rich systems that arise during climate change, allowing them to persist where climate is suitable. However, if intrinsic limits to alternative prey selection influence the owl's diet, then prey-rich systems provide the greatest opportunity for exploitation and persistence. Also, the continued presence of apex predators like the mast owl may be favorable because of the stabilizing effect of top-down pressure, which could mitigate some of the impacts of transient novel communities on ecosystems that can form during climate change. So using the combination of existing data resources and supplementing with field data, we updated knowledge on the distribution of mast owls and the potential impacts of climate change, highly valuable information for conservation in the future. So these previous 4 chapters with varying success have provided new data or knowledge on the mast owl. However, despite my best efforts, they remain data deficient. For my final chapter, I explored the impact of uncertainty in threat assessments. Which brings us to chapter 5. Can population viability analysis be a useful tool for evaluating the status of data deficient species? Rodenticides are a group of poisons used in rodent control that act on blood coagulation pathways and are non-specific, affecting a wide range of non-target species. This is a problem for predators that consume contaminated rodents. Secondary poisoning and bioaccumulation within predators is an unintended but serious conservation issue. In exploratory analysis, I found that mast owl carcasses contained rodenticides at harmful levels as indicated in this plot here. This is a good example of uncertainty in assessing threat impacts because the effects of exposure on the population may be substantial, but evidence for their specific effects is weak. Consequently, regulators appear reluctant to act on exposure data alone, meaning these products remain available over the counter at supermarkets and other outlets. From literature that includes a related species, the barn owl, the use of rodenticides can lead to decreases in breeding performance and increases in mortality. We wondered if we could use quantitative methods such as population viability analysis to explore the effects of uncertain knowledge in scenarios of rodenticide exposure. PVA allows the simulation of population trajectories under user-defined ecological scenarios, whilst quantifying the impacts of uncertainty in the data. We used the software Vortex to run PVAs. We first defined the demographic parameters for the mast owl. We then ran sensitivity tests to explore variation, and then defined scenarios where rodenticides impacted on population parameters. We defined the life history parameters from the small amount of literature available for mast owls, plus new information from my preceding studies. Specifically, I defined a single population based on the results of my genetic study, and likewise was able to ignore the effects of inbreeding based on a reasonable effective population size of greater than 500 and a non-concerning inbreeding coefficients. However, where data was completely unavailable, we used literature on the barn owl or left values at default. There was absolutely no data available for juvenile mortality in our search, so we estimated it at 54% based on literature for a variety of other raptors. A recent review of 160 PVA studies covering 144 species found that only 18% of studies were of high quality based on a range of metrics, including the underlying reliability of life history information. The best studied species, of course, tended to have the better quality PVAs. However, even the best PVAs can have missing data, meaning that some guesswork is usually required. Given the ubiquity of uncertainty in PVAs, most PVA software incorporates options for explicitly modeling uncertainty. In the case of the Vortex software, this is in the form of a sensitivity testing option where users can specify ranges for uncertain parameters. Then, in a Monte Carlo simulation, the full range of variation across all uncertain parameters is evaluated in terms of impacts on the simulated population sizes. Most PVA parameters can be included in sensitivity testing, giving the user great flexibility. Most of our parameters were not estimated from systematic field research and is therefore highly uncertain. We used a sensitivity test to simulate the effects of the extremes of 7 uncertain parameters listed here in the table, along with the ranges speculated. We used linear models and model averaging to determine that the most impactful variables on simulated population size came from variation in annual mean brood size and juvenile mortality and the interaction of the two. As you see in the figures, increasing brood size positively affected simulated population sizes, and increasing juvenile mortality negatively affected simulated population sizes, and the interaction of these effects is shown in the bottom panel. Given the importance of juvenile mortality in the sensitivity tests, for our rodenticide scenarios, we started with 3 base models indicated here in red text, featuring low, medium, or high juvenile mortality. To assess the impact of rodenticides, we defined 7 scenarios within each of these base models where plausible consequences of rodenticides on combinations of brood size, adult and juvenile mortality were compared. This was achieved in Vortex by imposing additional variation above or below baseline values to mimic known impacts of rodenticides on barn owls. This figure on the left shows the resulting population sizes and standard deviations after 50 years of simulation, and the table shows the results of pairwise contrasts of each of the base models with the 7 rodenticide scenarios with significance in bold text. When base juvenile mortality is low, the red bars, we see that the greatest single effect on population sizes came from impacts in brood size, the yellow arrow. However, not quite significantly, the red box. Whereas, when there is an impact on mortality in addition to brood size, then populations collapsed, yellow arrows here. Therefore, rodenticides resulted in the lowest population sizes only when multiple parameters were affected and only if the impact included an impact on brood size. It follows then that in the moderate juvenile mortality scenarios, the green bars here in the figure, the effect of rodenticide on brood size caused populations to collapse across the board, indicated with yellow arrows, and significance indicated with red boxes. The effects of greater mortality were not significant in isolation, indicated here with yellow arrows and red boxes. However, a dual impact on adult and juvenile mortality, the blue arrow, significantly lowered simulated population sizes, the significance indicated there in a blue box. Higher juvenile mortality models, the blue bars, were never viable regardless of rodenticides. So that mast owls were exposed to rodenticides is clear. The simulated impact of rodenticide on mean annual brood size had the greatest effect in our models. However, assessing this impact of rodenticides on mast owls was hindered by the uncertainty surrounding juvenile mortality. Still, rodenticides are just one of many threats, with others including habitat loss and degradation from logging, agriculture and urbanization, collisions with vehicles and other artificial structures, and competition for resources with other species. The impacts of these processes likely vary in space and time and act cumulatively on the population. However, there's only weak evidence of the impacts of these processes individually. Nevertheless, this study shows an effective approach to quantitatively model a theoretical range of impacts from speculative threats. So I used PVA to assess the impact of a likely but poorly understood threat to mast owls, whilst explicitly quantifying the effect of uncertainty in an important demographic parameter, juvenile survival. The study is a demonstration that even though our best efforts to reduce data deficiency fell short, this doesn't have to equate to conservation paralysis. Instead, techniques like PVA can be used to assess a range of possible scenarios whose outcomes can guide action until better information becomes available. So let's recap the chapters quickly. I used the Mast owl as a model of data deficient species and tried various approaches to improve its conservation. First, I attempted to obtain new data using an occupancy field study. I found that Mast owls were not widespread in our forest that has been shaped by decades of logging. However, my estimates of occupancy lacked confidence, and so I decided to pursue alternative approaches. Next, I found that traces are a promising source of DNA for population studies, but the rarity and difficulty of surveying for them could limit their application. Somewhat disillusioned with fieldwork, I turned to existing data sources. Using museum collections, I showed a declining effective population size over 6 decades, a single conservation unit, however, with isolation by distance. I then used occurrence records to show the impacts of climate change on prey distributions could influence the range of the Mast owl. And finally, I incorporated the new genetic information into a PVA where I simulated the effects of data uncertainty on a range of conservation scenarios that showed that juvenile mortality and variation in fecundity are critical determinants of extinction risk in threat scenarios. This last step shows how we can use demographic models to prioritize areas of uncertainty to inform adaptive research and monitoring for data deficient species. To conclude, many species are difficult to study and therefore data deficiencies are ubiquitous in conservation. As a bird, the Mast owl is amongst the best studied taxa, whereas many other taxa such as amphibians lack the benefits of well-established survey techniques, public interest, and the availability of collections. Consequently, our flexible approach to seeking new information by a variety of means to find what works best could apply to other taxa, as there is unlikely to be a one-size-fits-all approach to reducing data deficiencies. Demographic models such as PVAs provide a way to unify disparate strands of life history data so that theoretical conservation scenarios can be tested regardless of data gaps. Therefore, whilst waiting for better knowledge, preliminary conservation assessments can be made in a quantitative way that addresses uncertainty, which is potentially an improvement on qualitative assessments which don't assess their own uncertainty. And that completes the thesis. I should thank all the wonderful people that have been involved. A special thanks to my supervisors, Diane, Ross, and Robert, to the amazing people of the Difficult Bird Research Group for all your faith and encouragement. A big thanks to Nicole and Zorro, our scent dog. We didn't quite hit our mark, but we gave it our best. Also to Phil, Amy, and Jason at the FPA. I might not have even gotten started without you. Thanks to Nick Mooney for his knowledge and help along the way. A big thanks to TMAG and QVMAG museums for providing access to collections, to the wildlife refuges and sanctuaries for access to your owls, to Peter Stronach and Taslandcare, thanks for the support. Similarly to Tasland Conservancy and the Bob Brown Foundation, to everyone who contributes to wildlife databases, all of the landowners, and to Susie, my wife, for being with me through it. And thanks to these groups for financial support throughout the process. That finalizes my seminar. Any questions?
Congratulations, Adam. Well done.
Oh, thank you.
Now, I'm hoping someone in the room will be able to field questions there because I can't see the room.
Sure, I can do that for you.
Yeah, thank you. And I'm assuming people will type any online questions into the question box and I'll read them out and Adam can answer them. I might, unless there's any questions in the room, I might get us started by just inviting Adam to reflect on, Adam, do you think that the failure to find owl pellets using the scent dog reflects on the efficacy of the scent dog method itself, or do you just think it was about how that method was applied to searching for owls?
I personally just think about that it was the way we applied it. Absolutely. I think it was far too targeted. And because we targeted it too specifically to sites, the rarity of those pellets in the landscape was just too easily missed.
Yeah, okay. So
we could have continued, me and Nicole could have continued, but I mean, by that stage, I mean, we got slowed down, so we had to sort of stop.
Yeah, and I guess
otherwise my PhD would have gone for 9 years.
So,
do we have any questions?
I think there's plenty of opportunities.
Yes.
I think. Oh, sorry.
Oh yeah, we do have a question from the room. We can go to that now if you wanted to. But sorry,
yeah.
I just yep. Okay. So this is from anonymous attendee. What purpose will finding the what purpose will finding the minimum number of SNPs necessary to align pellets to individuals serve? So study 2. Is there any way to use the same SNPs across the sequencing runs as markers?
So the purpose of me doing it is simply because I pretty much answer both questions, I think, in the same run. So I think because I use a DartSec protocol, no, using the same SMP across runs, that 150 SMP set was not my goal, although I know that that's quite often the goal in forensics. Instead, I would say that I use a DartSec protocol so that each time you do one of those experiments, your results are comparable. However, you may end up using a different set of SMP. I shoot for the lowest set because there's so much missingness. So that between across samples, you have up from, what was it, 1.3 to 85% missing data. So you kind of got to shoot for a very small number of loci so that you've got at least some overlap between specimens. Does that answer your question?
We would hope so, because their mic isn't up. So speak up if you did want to, discuss that anymore. We can go to Phil Gibbons. He had a question to ask.
Oh, thanks. Can you hear me, Adam?
I can.
Yeah, look, that talk was really fantastic. I found it really clear. It was really brilliant. You've done some great stuff. And look, I was just wondering, when you're comparing historical and contemporary effective population size, is it feasible to break that down by land use if you had enough data?
I presume that yes, if you had enough data, but you'd also need to have enough samples.
That's what I meant, sorry.
Yeah. Absolutely. You can find that in the literature for sure.
Because wouldn't that be great to compare, land clearing and production forestry in Tasmania and how that's affected, changes in population size?
Yeah, it's pretty interesting though, because a lot of things can affect NE from year to year, like say behavior can definitely affect NE year to year, because it might affect the different percentages of males and females breeding and the percentage of the population breeding in any given year. So I think you could do it and you'd get some numbers. Figuring out a very sound inference might be difficult.
Okay, all right. Thanks. No, fantastic talk.
Okay, so we've got a question from Saul. Are there alternative rodenticides, which are harmless to owls, that have good prospects for wider adaptation?
There actually are, and I can't name them off the top of my head. And I forgot to put the citations at the bottom of my talk there. So the most of the research on barn owls is actually to do with seeking alternatives that are very active against rats, but have less of effect against the barn owls. So there's definitely alternatives there that are very effective against rats, but of course are a lot less safe around raptors and all of the other wildlife that are being affected by these things now. So yeah, definitely alternatives.
Okay, there's a question from Duddy Man. Adam, fantastic presentation. Thank you. What a journey you've been on. Interesting to see that your future climate models are a reasonably good match to the elevation profile of Tasmania. Was elevation one of the parameters of your models? And if I am interpreting your maps correctly, can you please bring them up on the screen again for this? It appears that MO disappear from Northwest Tas in many of these future climate models.
Yeah.
Why do you think that is?
Let me bring it up for you. Let's just go to the basic one. That should bring it up. So you're talking about Northwest, why it disappears from Northwest Tas.
I'll read that last bit. Yes. Okay. The answers come through as yes. That's what they're asking.
I think that in a lot of the models, the values over in the Northwest there just actually start going outside the model, the range of variables that we use to produce the model. So you get that effect. But I think more it's just being driven by the fact that that west still remains somewhat wet. And within the models, it's the drier and higher temperatures within the climate variables that are actually driving these predictions into the east. So it's not so much the changes in the Northwest that are doing it as it is the changes in the east that are doing it. Does that make sense?
As much as SDMs make sense to any non-specialist, I think.
Oh, sorry, because yeah, I guess I didn't explicitly tell you all which bioclims were used. So I didn't use elevation. I use because it conflicts with too many of the other bioclims, right? So you end up with huge multi-collinearity problems. So for me, the cool temperatures essentially were a surrogate for elevation. Now, what I'm saying is that precipitation contributed very strongly negative, and mild to higher temperatures contribute very strongly to the positive of a prediction of strong occurrence of Mast hours. So the drive into the east is more to do with the drying and warming of that area and less to do with the changes in the Northwest. I hope that makes sense.
Yeah, the answer was yep, thanks. So
okay.
And Ross Crates wanted to say excellent presentation and thesis, Adam.
Ross.
Congratulations on this important work on a very difficult bird. We didn't have any other questions coming through. So if anyone has anything else, now is your chance. Otherwise, I'll probably hand it over to you guys to wrap up.
Yeah, I can't see anything else coming through online, and we're at 2:59. So I guess we'll just take this moment to congratulate Adam again on a thesis, hard one and well done, despite multiple challenges, not least of all COVID, and you should be very proud. I think you've done a great job. Congratulations.
Thanks.
All right, thanks so much, Adam.
Yes.
I'll close off this webinar now, and and a congratulations from Saul as well. So there you go.
Thank you, Saul.
Thanks everybody for attending.
Okay, thank you.